Good evening. ARVO is privileged tonight to participate in recognizing two outstanding vision scientists who received the Antonio Champalimaud Vision Award during this past year, Jeremy Nathans and Qinghua Yao. This is the second year of the Antonio Champalimaud Vision Award. The award has a dual nature. It is given to recognize contributions to the alleviation of diseases affecting the eye and vision, particularly in developing countries. And in alternate years, as this year, it recognizes contributions to overall vision research. The recipients of the award are productive research groups rather than single individuals. This may involve groups from more than one institution or discipline. The Champalimaud Foundation is based in Lisbon, Portugal. The foundation was created through the bequest of the late Portuguese industrialist and entrepreneur, Dr. Antonio de Summer Champalimaud. As stipulated by Mr. Champalimaud, prior to his death, the foundation's president is Leonor Beleza, former Portuguese Minister of Health. We are joined tonight by a member of the Champalimaud Executive Committee, Dr. Antonio Borges, former Vice Governor of the Bank of Portugal. The Vision Award jury consists of an international panel of outstanding vision research scientists, and they are joined by exceptional public figures who are involved in meeting the needs of the developing world. The panel includes two Nobel Prize winners. This award is one of the largest bestowed on an annual basis. Currently, the award provides €1 million to the recipients for their use in any way that will further their studies and contributions to their scientific and humanitarian endeavors. Last year, the 2007 inaugural Champalimaud Vision Award was given to the Aravind Eye Care System in Madurai, India, to recognize their remarkable contributions to global eye care for indigent people and for their work in global prevention and amelioration of vision loss. Last year at the 2008 ARVO meeting, we heard a moving presentation by Dr. Nam, CEO of the Aravind Eye Care System. This year marks the second Champalimaud Vision Award. The award is to recognize discoveries that enhance our knowledge and understanding of vision. It goes to two exceptional vision scientists, Jeremy Nathans and Qinghua Yao, who have this in common. They have developed a deep understanding of the biology and function of the photoreceptor cell, which is the first cell in the neural chain of vision. In a moment, we will be privileged to hear from both of these recipients of the 2008 award. I would alert the audience that nominations during the coming year are toward the selection of recipients for outstanding basic and clinical vision research again. Let me just briefly introduce the two awardees. Jeremy Nathans has made fundamental discoveries in basic and clinical science of the photoreceptor cell. He isolated the genes that encode the human visual pigments and defined the molecular and biochemical basis of inherited variation in color vision. He elucidated the biochemical basis of multiple inherited retinopathies, and he discovered unexpected mechanisms that regulate rod and cone photoreceptor development. Dr. Nathan's research has illuminated a broad landscape in vision using the tools of genetics, molecular and cellular neurobiology, and developmental biology and ophthalmology. Qinghua Yao has made major contributions also to elucidating photoreceptor mechanisms, particularly the light responses of rod and cone photoreceptors, and to solving the underlying phototransduction mechanism, including establishing that cyclic GMP is the messenger for photoactivation, and that calcium is a critical messenger for adaptation. He also contributed greatly to understanding the intrinsically photosensitive retinal ganglion cells, which are non-rod, non-cone photoreceptors that mediate circadian photoentrainment and other non-image forming visual functions. Among other discoveries, he found that vitelliform macular dystrophy, also known as Best's disease results from mutations of a new chloride channel protein, and his work provides significant steps toward uncovering fundamental molecular mechanisms of vision. Let me then introduce Qinghua Yao, who will make his presentation. Thank you.
I would like to thank Dr. Paul Sieving for the very kind introduction, and to thank the jury on behalf of which Paul has just spoken of. And I would also like to thank the Champalimaud Family and Foundation for recognizing the importance of vision research and fostering it. Most of my work for the past almost 30 years has been on how light triggers vision. Now, by the early 1980s, there were already many important observations about this process in retinal rods and cones, but there were also puzzles and controversies, especially on whether and how calcium and cyclic GMP partake in phototransduction. I was fortunate to be in the right place at the right time and managed to put these controversies and puzzles into a coherent and logical picture. For a diverse audience like this one, please let me just briefly summarize this picture. So this is a picture of a rod photoreceptor. Its outer segment contains the visual pigment and transduces light into an electrical signal. And the way this is done is as follows. In darkness, a current flows into the outer segment through open cation channels on the surface membrane. This current depolarizes the cell and maintains a steady release of glutamate from the synapse. Light closes these cation channels, therefore stopping the inward current. As a result, the cell becomes hyperpolarized, and this hyperpolarization reduces or completely stops the glutamate release. Now, these events at the outer segment had long been established by Shunil Tomita's group in Japan and Bill Hagen's group at NIH. Likewise, the events at the synapse were established by John Dowling, Harris Rips, Luigi Chiavetto, Akimichi Kaneko, Bill Pack, and others. It is the question of how light closes these cation channels that had long remained controversial. The final picture, which has remained essentially unchanged since 1985, is as follows. So this is a cartoon picture of the rod outer segment. This is the surface membrane. And these are the light-sensitive cation channels open in darkness and closed in the light. Yevgeny Fesenko's group in Russia were the first to report that cGMP activates a cation channel on the rod outer segment. Our lab then showed that these cGMP-gated channels are one and the same as the light-sensitive channels mediating vision. These are the cyclic GMP metabolic reactions catalyzed by the cyclase and the phosphodiesterase. Light photoisomerizes rhodopsin into an active form, which then via activation of the G protein transducin by GTP-GDP exchange stimulates the phosphodiesterase to increase the hydrolysis of cyclic GMP. As a result, the level of cyclic GMP falls and the channels close to produce the light response. The essential biochemistry of this activation process was worked out in the 1970s and early 1980s by Mark Betensky, Paul Liebman, Hermann Kuhn, Lubis Stryer, and others. There are several deactivation mechanisms. First, the active rhodopsin is phosphorylated, followed by the binding of the protein called arrestin that caps the pigment activity. Next, the transducin deactivates itself by endogenous GTPase activity, which hydrolyzes the bound GTP to GDP. This GTPase activity is facilitated by a gap complex consisting of the protein RGS9, its associated protein R9AP, a previously orphaned G protein beta subunit called G beta 5, and finally the gamma inhibitory subunit of the phosphodiesterase, which in fact is a substrate of transducin. When transducin deactivates, so does a phosphodiesterase, because the gamma subunit resumes its inhibition on the catalytic alpha and beta subunits of the enzyme, therefore shutting off transduction. Consequently, cyclic GMP goes back to the dark level because of ongoing cyclase activity, and the channel reopens. In other words, at all times, there is a steady synthesis and hydrolysis of cyclic GMP, and light simply tips the balance by increasing hydrolysis. Many of these deactivation details, some still ongoing, were worked out by various people, including, if you don't mind, I'm going to go through the list: Hermann Kuhn, Paul Liebman, Derek Bounds, Vadim Ashavsky, Ted Wenzel, Jim Hurley, Mel Simon, Dennis Baylor, Jeannie Chang, Jason Chang, Clint McKinnell, Marie Burns, Fred Rickey, Trevor Lamb, Ed Pugh, and others. It is quite a crowd, but a very good crowd. Calcium also plays a very important role in phototransduction. In the early 1980s, we discovered this circulation of calcium at the outer segment membrane, consisting of an influx through these channels and an efflux through this sodium-calcium exchange mechanism, which Peter McNaughton later showed to be also dependent on potassium. In the light, the calcium influx stops because the channels close, but the efflux continues, so the net result is a decrease in the free calcium concentration in the rod outer segment. Now, we ask the question, what happens if we prevent calcium from falling? So there's actually a very simple way to clamp calcium in the outer segment, which we did, as the Trevor Lamb, Gordon Fain, and co-workers did. And what happened was, when you prevent calcium from changing, the rod essentially loses all its ability to adapt to background light. Then from the 1980s all the way to the present, a lot became known about what calcium is doing inside the rod outer segment, as shown by these red arrows. There are several feedback mechanisms. One is on the cyclase, a second is on the phosphorylation of rhodopsin, and a third is on the activation of the channel by cyclic GMP. Likewise, quite a few groups have worked this out, including Satoru Kawamura, Dick Lawley, Lupus Stryer, Bob Moday, Jim Hurley, Chris Palchevsky, Wolfgang Baer, and others. In the mid-1990s, our lab decided to dissect in detail these feedback pathways, and we found a very interesting conclusion, namely that there's a division of labor between these feedback mechanisms. In low light intensities, we found that this feedback on the cyclase is the predominant one in determining the sensitivity of the rod cell. As the light intensity continues to increase, this calcium feedback on the phosphorylation rhodopsin begins to kick in to facilitate the adaptation of the rod to illumination. This feedback on the channel, however, appears to be weak and unimportant at all light intensities. Incidentally, for a long time, in fact from about 1960 to about 1990, there was a myth in the field, and that is that it was thought that mammalian rods, unlike amphibian rods, do not actively adapt to light. We spent about 3 years in the late 1980s to about 1991 sort of screening many mammalian species from rat to rabbit all the way to various primate species, and we found in fact that all of these photoreceptors adapt to light. In fact, they behave in such a stereotypic fashion as if the rods are already optimized in their function. And when a mammalian species need to stretch its dynamic range in terms of light intensity, all the animal does is to sprinkle more or less cones in the retina to deal with that. Now, as an aside, I may also mention that rod phototransduction, being so well understood, has provided really it heralds the understanding of many other G protein signaling pathways, of which there are hundreds and thousands in the body. In particular, olfactory transduction that mediates the sense of smell has a mechanism very similar to what is shown here for rod photoreceptors, including the existence of these calcium feedbacks, except that we have found that this feedback on the channel is actually very potent in olfaction, although it is rather weak in vision. So it's kind of an interesting difference in emphasis of mechanisms between the two senses. Finally, the study of the structure and function of these cyclic GMA-gated channels has literally burgeoned into its own field of research, beginning with the cloning of the A subunit by Benjamin Kauf's group. Soon after, we discovered the B subunit also by cloning, and much later, in collaboration with Bob Moday's group, we solved and found a rather unusual asymmetric stoichiometry between the A and B subunits in a native rod channel complex, apparently to ensure a specific and selective assembly of these channels in the rod cell. Now, what about cones? Well, again, in the early 1980s, there was uncertainty about whether cyclic GMP plays any role at all in cone transduction. However, our lab, and separately at Pugh's lab, have shown that indeed and affirmed that cGMP does play a key role in cone transduction. So this picture here applies perfectly to cone transduction as well, even though there are quantitative differences between the two kinds of photoreceptors. More than that, most of the proteins involved in transduction have different isoforms in rods and cones. Now, with this picture of transduction being so well understood, in fact, every gene has been cloned, the ones shown here and many not shown here, and many of them, when mutated, are found to cause a disease or another. So with this mechanism so well known, the hope is that eventually some treatment can be made for many of these diseases. Now, in a separate direction, because of my broad interest in vision, I have also decided to follow the footsteps of Gus Amberson and Eduardo Solasio and look at a rather unusual eye called the parietal eye, or the third eye, which is present in lower vertebrates. This is a bird's eye view of the forehead of a lizard, and the parietal eye is situated right here at the top. Now, the photoreceptors in this eye are very much like rods and cones. In fact, they are also of a ciliary nature, except, which is very unusual, is that each cell here expresses two pigments, and there is color opponency within a single cell. In the late 1990s, we solved the transduction mechanism and the color opponency in this cell. And more recently, we've cloned the major components involved in the transduction mechanism. And what came out of that investigation is kind of interesting. It appears that these cells are an evolutionary missing link between the ciliary photoreceptors in invertebrates and our rods and cones. Because of my interest in a broad perspective in vision, we also went on, in fact, venture in the newly discovered melanopsin photoreceptor system in the retina. Now, the existence of a non-rod, non-cone photoreceptor system was first demonstrated by Russell Foster, Robert Lucas, and co-workers. The search for this photoreceptor was then accelerated by the discovery of the pigment melanopsin by Iggy Provencio and Mark Rolak. And finally, it was unequivocally identified by David Berson's group as a population of intrinsically photosensitive retinal ganglion cells. David Berson's lab and our lab then joined forces to show that these intrinsically photosensitive ganglion cells indeed express melanopsin. So this is a flat-mounted retina, red retina, and the green color is the melanopsin immunofluorescence, which is present all over these cells, the cell body, the dendrites, and even the proximal part of the axon. And these cells number about 1,000 per retina. Well, in a knockout animal we made that lacks melanopsin, these cells are no longer light sensitive. Furthermore, in a triple knockout animal that we made that lacks rod or cone functions and also no melanopsin, the animal loses all its ability to detect light, whether for conventional vision or for accessory visual functions such as circadian photoentrainment and the pupillary light reflex. Incidentally, Sachin Panda, Iggy Provencio, Russell Van Gelder, and others have obtained similar results. Now, in the melanopsin knockout mouse, we actually replace the melanopsin gene with a tau-lexi marker gene. This allows us to visualize all the axons coming out of these ganglion cells, as shown here by the blue color, in this case in the retina, going to the optic disc. We can trace these axons all the way to the brain, and in collaboration with David Berson again, we were able to identify all the nuclei in the brain that receive innervation from these cells, prominent among which are the suprachiasmatic nuclei, which is the primary circadian pacemaker in the brain, and also the olivary pretectal nucleus, which drives the pupil constriction. In collaboration with Dennis Dacey and Paul Gamlin, we found the same melanopsin system in the primates, including our own eyes. So it looks like that this system is conserved across all mammalian species. Most recently, our lab has carried out a detailed biophysical study of these retinal ganglion cells, and rather surprisingly, we found that these cells actually have very little melanopsin pigment on them, perhaps to avoid interfering with the passage of light to the rods and cones lying underneath for image vision. On the other hand, even though the photon capture is very, very low, these cells are extremely efficient in that they can signal the absorption of a single photon to the brain, just like our rods. Finally, let me mention that as we go down to lower vertebrates, the concept of which neurons in the retina are intrinsically photosensitive becomes even blurrier. Most recently, we have found that even after dissociation into single cells, many fish horizontal cells remain photosensitive. And this effect of light appears to be a modulation of the voltage-gated calcium current. Well, where we're heading from here, we're still working on rods and cones, and we're still, of course, interested in the melanopsin ganglion cells. However, being forever restless, I always like to push the envelope and venture into territories not covered by the NIH grants. So I think the funds from the Champalimaud Foundation would come in just very handy. But it must be said that my research over the years has been and remains to depend on the National Eye Institute. And I would like to take this opportunity to thank the National Eye Institute for all the support over the years. In closing the talk, I'd like to acknowledge two mentors in vision research. The first is Dennis Baylor, who has taught me a great deal and who remains my closest friend. Dennis and I worked together side by side to develop the suction pipette recording method, which, although unintended at the time, turns out to be very useful for understanding phototransduction. Next, actually, more than 30 years ago, there was one memorable afternoon when the two of us were able to observe the response of a rod to a single photon for the very first time. Second is Alan Hodgkin, a scientific giant whom I had the great fortune to work with. And Alan taught me one thing, which is how to think. I also would like to thank Jeremy Nathans for a wonderful collaboration on discovering a new class of chloride channels, a member of which, when mutated, causes the human Best disease. And finally, these are all the people who have worked with me in the past and the present. Without them, obviously, nothing would have come out of the laboratory. And so this award belongs to them as well. Thank you very much.
Thank you. Brilliant. Thank you. Congratulations. Thank you, King, for a superb elucidation of the function of photoreceptors over many years. I would like all of us now to welcome Jeremy Nathans to continue the conversation, perhaps in other forms.
Great. Thank you, Paul. Well, thank you very much, Paul, for hosting this session. It's a pleasure to be here. I want to thank the Champalimaud Foundation for putting their faith in King and myself and putting their faith in vision research. I think you've made an enormous difference, and there's much to come because the foundation is going to have a very profound effect on vision care and vision research throughout the world. So thank you very much. Now, I want to start in telling you this story about one ongoing research project that we are very excited about by perhaps starting at the end, which is to show you the people who've done the work, and I want to mention them as we go along. In my lab, Yanshu Wang, Xinyi Chengzhu, Phil Smallwood, Hugh Cahill, John Williams, and Tudor Badia have been the main movers, and we've had a series of wonderful collaborations over several years with Minjong Yu, Neil Peachy, Alain Dabdoub, Chad Woods, Matt Kelly, Bill Tasman, Li Jiang, and Kang Zhang. And really it's been a collective effort, and I want to echo what has been said before, which is that the Champalimaud is really recognizing an entire research group, not just the head of the lab, and that's very much the case here. So the story I want to tell you about relates to vascular development in the retina, and it begins with the discovery in the 1980s of an unusual family of receptors called frizzled. This was first found in Drosophila. They look like G protein coupled receptors. There are 7 transmembrane segments shown schematically on the bottom. There is a extracellular N-terminal cysteine-rich domain shown at the top. The original frizzled family member was identified in flies as a gene which has an unusual cuticular phenotype. And we discovered, somewhat by accident I should say, a family member in the human retina, expressed in the human retina. And when we read the papers from the fruit fly world, we thought that there were some interesting things to be discovered potentially in mammals, so we decided to follow it. To make a long story or a series of stories short, there are 10 frizzled family members in mammals, and the frizzleds we showed in the 1990s are in fact the receptors for a large family of ligands called Wnt. There are 19 Wnt in mammals. And the one I'm going to tell you about today, frizzled 4, has a particular role in the retina. I don't think I have to tell this audience that the retinal vasculature is important. It's obviously important clinically because there are many retinal diseases that are in essence vascular diseases. It's also, it looms very large in vascular biology, in part because it's the only vasculature in the human body that can be imaged non-invasively. And of course, as you know, this has been examined through the ophthalmoscope for over a century and a half. And so there's a vast literature on the biology of the retinal vasculature, more than any other vasculature by far. Now, there are 2 particular diseases that caught our attention, and they're mentioned here, and in particular the molecular genetic underpinnings of these diseases are mentioned here. They are Norrie disease and familial exudative vitreoretinopathy. Norrie disease is an X-linked trait. In the early 90s, the gene was identified, and it was found to encode a small protein. It's a secreted protein, and it has the same cysteine knot pattern that the TGF-beta family has. So it's a ligand of some sort. And fever, which, like Norrie disease, is a hypovascularization disease, and Norrie is more severe in general than fever, but both have that same hallmark of insufficient vascularization, is genetically heterogeneous. There are a variety of forms, but in the early part of this decade, it was found that one form involved mutations in frizzled 4, and a second involved mutations in the co-receptor for frizzled 4 called LRP5. So frizzleds actually work not just alone, but with a co-receptor protein. Now, we got interested in this business in particular when we knocked out the frizzled 4 gene in mice, and we found this spectacular vascular phenotype. So on your left is the normal retinal vasculature. This is a flat mount of a retina, and we've filled the intravascular space with fluorescinated dextran. You can see the large vessels, and then there are several tiers of capillaries at the vitreal surface and on either side of the internuclear layer. And in the frizzled 4 knockout, the large vessels are present, but there are no capillaries within the retina. They just have these little nubs, and here in cross-section you can see what this looks like perhaps a little more clearly. In the wild type, here's the ganglion cell layer at the bottom, outer nuclear layer at the top. You can see the inner tier of capillaries, one here and one here. And in the mutant, there's sort of an excessive abundance of vasculature vessels on the vitreal face, and then there are these just blind endings of cells that have failed to make intraretinal capillaries. Now that is a striking phenotype, and it's also striking that exactly the same phenotype was seen by Wolfgang Berger's group in Switzerland when they knocked out the Norrie disease gene. And so that got us to thinking, even though the Norrin protein, the product of the Norrie disease gene, has absolutely no homology to Wnt, the canonical ligands for frizzled, that maybe that Norrin protein is in fact a ligand for frizzled 4, at least in this context. And that guess turned out to be right. So I'll show you one experiment, or a couple experiments that demonstrate that. Here we are looking at a luciferase assay, a reporter cell line that signals canonical Wnt signaling. And we have transfected those cells with frizzled 4. The row is just a certain rim or epitope tags. You can ignore them. Frizzled 4 and Norrin, and one of the two essentially equivalent co-receptors, LRP5 or 6, highly homologous proteins. If we have all three, as shown here by the plus signs, we get very robust signaling. If we have only two of the three, it doesn't matter which two, you get very little signaling. And either co-receptor will work. So here we've done it with LRP6. So these work in combination to affect cell signaling. And furthermore, Norrin binds with nanomolar affinity to frizzled 4, and it binds with exquisite specificity. So here's a set of binding experiments in which cells have been transfected with the different frizzled ligand binding domains, frizzled 1 through 8, 9, 10, and a series of 5 frizzled-related proteins. These are decoy receptors made by other genes. That's the sum of all the frizzled genes in the genome and the sum of all the frizzled-related proteins in the genome. And then here on the left set of panels, we have probed those cells with an anti-MYC antibody just to demonstrate that the proteins are expressed on the cell surface. They are. And on the rightmost set of panels, we've probed with a Wnt protein, happens to be Wnt8, and it's alkaline phosphatase fused, and we can see which frizzleds bind to it. Turns out to be 4, 5, 7, and 8. And that's fairly typical for Wnt. They tend to be promiscuous in their binding. But now if we probe with Norrin, it's a very striking result. The only receptor that binds Norrin is frizzled 4. It's a private ligand for frizzled 4. Now, that set of observations begs a number of questions. For example, which cells in the retina are using the signaling system? It also begs the question whether the system is used outside of the retina. And in fact, there is an inner ear vascular phenotype in both the frizzled 4 knockout and the Norrie disease knockout mouse, which is because there's a progressive degeneration of the stria vascularis, so there is some action outside of the eye, but we were wondering more generally, is there signaling throughout the vascular system beyond that? And also one would like to know what are the consequences for visual function of defects in Norrin frizzled 4 signaling? And finally, what's the molecular underpinnings of that signaling downstream? Okay, so the first step, where is the signaling happening? So Xin Yi, a grad student in the lab, made a conditional knockout mouse, and the business part of this slide is down here. Frizzled 4 is conveniently a small gene, so she could put a LOX-P site just upstream of the methionine codon, another LOX-P site just downstream of the transcription termination site, and then that would be a conventional conditional knockout. But then she put an alkaline phosphatase coding region, this is a plasma membrane protein, downstream of the second LOX-P site. So when you recombine this allele, you eliminate frizzled 4 between the two LOX-P sites, and alkaline phosphatase is now turned on. It wasn't before, because it wasn't transcribed, is now turned on under the control of the frizzled 4 promoter. And you can use that to determine where frizzled 4 is expressed. So, for example, if we recombine that conditional knockout over a wild-type allele with a endothelial Cree, we see the entire vasculature light up in this E11 embryo. And as it turns out, frizzled 4 is expressed in many places. I'm not showing you all of them. But it is expressed in all of the vasculature all the time, from the very beginning to the very end. Furthermore, we can put this conventional knockout over the conventional knockout over the conventional null allele, the original null allele, which doesn't make alkaline phosphatase. And then we can ask, if we eliminate function in only the endothelial cells, and that's shown in this lower image here, do we get the phenotype? And the answer is yes. We get the full-blown phenotype. Here's a flat mount. All the endothelial cells are alkaline phosphatase positive, hence the black stain, and they are missing the intraretinal capillaries. So this answers one question, which is the critical cells for signaling are the endothelial cells. Now, I'll just say parenthetically, this technique here, I've shown the alkaline phosphatase staining in red, is especially useful for mosaic analysis. I won't go into detail, but just to say that since we can visualize individual mutant cells because of the histochemical marker, we can look at their behavior in a surround that's otherwise wild type. Highly useful. So, for example, we can see that we can make individual mutant capillary endothelial cells. Once the capillary bed is formed, that seems to be okay. The cell can be made frizzled 4 minus. It just can't make capillaries at the beginning, as shown by this cluster of clones, which have the characteristic balls of cells that have failed to penetrate the retina to make capillaries. Now, what other things can one say about the retinal vascular phenotype? Well, it's more than just a capillary defect. There's a defect in the growth of the vessels across the surface of the retina. They tend to be retarded. So here's a mutant retina at postnatal day 7 compared to a wild type. In the wild type, the cells have gotten already to the edge of the retina. In the mutant, they're only about halfway there. There's also a paucity and sort of an impoverished branching of the vascular network. So there's a whole series of defects here. There's also a severe defect in the interaction between endothelial cells and pericytes, the mural cells. So, for example, if you look at a postnatal day 2 retina, when the vasculature is just beginning, so this is not a secondary effect of later events, as I'll show you in a second here, the endothelial cells in green and the pericytes in red are right up next to each other. They're extremely closely connected. And in the mutant shown here, you can see that the red and green cells are really not particularly good at talking to one another. There are plenty of pericytes that are not touching endothelial cells and vice versa. Finally, there's one more defect I want to mention, just sort of an intrinsic defect, and that is that frizzled 4 signaling seems to be important in maintaining a blood-brain barrier. It's certainly defective in the retina. It's also defective outside the retina in some particular places, and in particular in the cerebellum. So if we immunostain a section through the cortex and cerebellum and probe with an anti-mouse IgG, so we're just looking at immunoglobulin, which normally, of course, should be intravascular. We see that it is. The green stain is confined to the vascular spaces in this neuron knockout mouse. So neuron is really not particularly relevant to vascular development in the brain. And it is confined to the intravascular space in this conditional knockout over wild type, because these would be now made heterozygous by the Tie2-Cre recombination. But in the conditional knockout over the null allele, the Tie2-Cre, that is the endothelial-specific Cre, eliminates frizzled 4 function in all endothelial cells. And although there's no effect on the cortex, which is shown at the bottom here, the IgG leaks into the tissue in the cerebellum. And there's this very impressive compromise of the blood-brain barrier in the cerebellum. Now, why is it so specific? I hope you're wondering, as we were scratching our heads, why, if frizzled 4 is in all the vasculature, in the brain, lungs, everywhere, why is the phenotype so specific? And here in the brain, why should it be cerebellum and not cortex? I don't really have an answer for that, except to say that we know that there's a lot of redundancy among frizzleds in their patterns of expression. We know there are other frizzleds in the vasculature, and we imagine that in this instance, the phenotype that we're seeing is just the tip of the iceberg. It's just that part which is not covered up by redundancy with other frizzleds. But the basic frizzled signaling in the vasculature will be universal to all vasculatures. That's an hypothesis at the moment. Now, let me just give you a little bit of evidence that suggests that might be right and addresses maybe more directly the role of frizzleds outside of the eye and ear and cerebellum. And so we asked, well, if we can't reveal frizzled function in other vasculatures by removing at least one frizzled, maybe we have to remove multiple frizzleds, maybe we can reveal it by overactivating frizzleds in various vasculatures. And we thought this could be done by taking advantage of the specificity of neurin. So since neurin is essentially a private ligand for frizzled 4, if we can express neurin, we can activate frizzled 4 selectively and no other frizzleds. And so we built, again, Shin-Yi built this construct in which neurin is controlled by Cre recombinase. There's a stop-locks upstream of it. This is a universal promoter, so it will express it everywhere. If we make neurin everywhere from early in embryogenesis, we end up with a lethal phenotype at mid-embryogenesis. And here's the mutant embryo. This is the embryos inside. This is the yolk sac here and placenta down here. Here's a wild type on the left. And you can immediately see not only is the embryo a bit smaller inside, but the vasculature of the yolk sac seems to be gone. I'll show you in a minute. It's not completely gone. Oops. When you look at the embryos, actually this is an embryo from the mutant. It's still alive. The heart was beating at the time we took it out. But the vasculature is completely disorganized, and that has led to a severe growth retardation. And if we look at the yolk sac, where we can see the vasculature very clearly, in the wild type you have this very distinct set of vessels, large vessels, medium-sized vessels, capillaries. The mutant is just this mass of homogeneous capillary-like vasculature. So kind of pleasingly, it's the opposite of the retinal phenotype. In the retina, the loss of function eliminated capillaries, and here in the yolk sac, the overexpression of neurin and over-signaling gave only capillaries. So it seems to be activating sort of a capillary development program. Now, how about question 3, the effect on the function of the retina, visual function. This was done in collaboration with Minjong Yu and Neil Peachy, and if you look at ERGs of rods and cones, what you see is that, unlike the wild type, there's absolutely no B wave. The A wave is quite normal, in fact. So there's a severe defect in intraretinal transmission. This is seen not just in the straight knockout, not just the straight knockout frizzled 4 minus. It's seen in the same complete form in the frizzled 4 knockout that is endothelial specific with Ty2-Cre. And a reciprocal experiment, if we eliminate here with RX-Cre, a homeobox that's expressed in all retinal neurons and glia, if we eliminate frizzled 4 in everything in the retina except for the vasculature, then we get a normal ERG. So it's really exclusively a vascular defect, even though frizzled 4, I should say, is widely expressed in the retina. It's in neurons and the vasculature. Now, when we looked with a bit more of a functional analysis, we saw something interesting. As you might expect, an optokinetic response, this is Hugh Cahill's work, which you can see in a wild-type mouse. This is the response to a moving grading of vertical bars, is eliminated in the frizzled 4 knockout or the frizzled 4 vascular knockout. But interestingly, the pupil response, the sort that King described just a few minutes ago, is actually quite close to normal. Not perfectly normal. Probably this failure to get a complete pupil response is due to the failure of rod and cone signals to reach the melanopsin ganglion cells. But the melanopsin ganglion cells seem remarkably functional as judged by this response. And so this was kind of striking. As judged by the A wave, the photoreceptors seem quite healthy. As judged by the pupil response, the ganglion cell layer seems quite healthy. So the vascular defect seems to be, in terms of its functional effect, largely limited to that central tier of neurons, the ones in the internuclear layer, a very extreme division of vascular territories, very, very precise division. And so we wondered, partly because the histology of the retina looks so incredibly normal, whether those cells might essentially just be dormant. That is, although they are starved for oxygen, perhaps they would come back to life if given sufficient oxygen. So we just took the retina out. This is Tudor Badia's work, and put it in Ringer's buffer, plenty of oxygen, plenty of glucose. It's sitting on an electrode, a multi-electrode array, and we just asked, does it respond to light? And the answer is, amazingly, it responds just as a normal retina does. So if you record ganglion cell responses, these of course are rod and cone driven, you see on responses, off responses, on-off responses, the whole gamut of ganglion cell responses. So here's an on response, off response, on-off response. So this is kind of intriguing from, I think, just the sort of the CNS disease point of view, that neurons can be essentially asleep, in this case, for months, but they're not dead. They can come back to life. And so there's a level of hypooxygenation which is required for function, perhaps, but not so severe as to kill the cells. Okay, now the last question. What's the molecular basis for signaling that confers the properties on these endothelial cells that frizzled 4 neurin signaling is important for? And so we've tried to answer this by stripping the system down to its most minimal components. And so knowing that signaling is critical within endothelial cells, Yanshu Wang did the following. She isolated the retinas from wild-type and mutant animals, dissociated the cells with proteases, and then immunoaffinity captured the vascular cells with an anti-PCAM antibody stuck to magnetic beads, put those in culture, and grew cell lines from both wild type, from LRP5, the co-receptor knockouts, from Neuron knockout mice, and from Frizzled-4 knockout mice. And I should say this is done with the so-called Immorto mouse background, where you can make stable cell lines from various primary cells. And in all cases, one gets cell lines that look quite normal with respect to their growth properties. They're staining with vascular markers. Here's vascular endothelial cadherin staining. So at this level, nothing seems to be wrong with the cells. But if you ask these cells to make capillary-like structures on matrigel, a classic endothelial cell behavior, you see a striking difference in behavior. This is 6 hours after putting the cells on matrigel. When you first put them down, they look like little balls, little billiard balls, and within hours they start to wiggle around, put out processes, and connect up in this network. Here's the wild-type cell, a wild-type cell line. Here's LRP5 mutant cells, which seem to do this quite well. We think probably because they're making LRP6, which we know they are by RT-PCR. The Nori disease cell line here can also do this, but the Frizzled-4, the receptor cell line, cannot. The cells just sit there and do nothing. Now, one might ask, maybe you're asking, is this because there's ligand present in the medium during this assay? I think there probably is. It's done in 10% serum, and there could well be WNTs that are activating the Frizzled-4. There may also be Neuron, for all we know. But in any case, this is a difference between them. There's kind of an interesting sociality to this difference. This is kind of a social behavior. And here I'll just show you the effect of mixing mutant and wild-type cells together. Mutant cells, as we just saw, just sit here like lumps on a log, doing nothing. Wild-type cells, labeled in red in this experiment, make these beautiful networks. But if you mix wild-type and mutant together, and here at different ratios, 3-to-1, 1-to-1, or 1-to-3 for mutant to wild-type, the mutant cells will participate in the networks formed by the wild-type cells. And so we think there's some sort of perhaps adhesive interaction or some sort of communication that the mutant cells are incapable of initiating, but are capable of responding to. And so they're listeners, but they're not talkers. And we'd like to know what that system is. Now, in terms of the molecular level, we've been looking at genes that are activated or repressed by Neuron Frizzled-4 signaling, done by microarray analysis with Affymetrix microarrays. And there are a number of ways we've addressed this. We've compared, for example, the yolk sac cells with the Neuron overexpression. So here we're asking if we activate the system, what genes get turned on and what get repressed. So this on the horizontal axis is the fold change in abundance. On the vertical axis is the p-value, because we've done this in every case in triplicate. So let's just look at the ones that are of high statistical significance. There are a bunch of genes activated, a bunch of genes repressed. Maybe not too surprising. Interestingly, if you look at the yolk sac at embryonic day 8.5, very, very early, when it looks completely normal, but it's already going down the road of now it's got Neuron overexpression and it's going to become abnormal, one set of transcripts that really stands out are those encoding SOX17, a transcription factor. These are upregulated by Neuron signaling. There are a bunch of other things of interest, TIMP3 and so on, we don't have time to go into. A similar analysis with the cells in culture also fingers SOX17 as an important player. So if we compare wild-type retinal endothelial cells in culture with mutant endothelial cells, again, we see that the wild-type make SOX17 much more than the mutant, and maybe more tellingly, if we stimulate wild-type cells with Neuron, as shown in this experiment to the right, where now we're looking at transcripts that have been stimulated on the right by Neuron or repressed on the left by Neuron, compared to the mutant cells, which Frizzled-4 knockout cells that lack receptor, where there's very little change in transcript abundance as a result of Neuron expression, essentially the same levels of transcript here at the 0 point. We see again, SOX17 has been activated in the wild-type cells. So this got us to thinking, among all the various genes that are controlled by Neuron and Frizzled-4, maybe SOX17, this transcription factor, which I should say has been implicated in zebrafish and in mice in vascular development in general, is a critical node in the communication of that signaling pathway to the nucleus. And so Shin and Yen Shu did a very simple experiment. They made a lentivirus that expresses SOX17, and they used it to make stable derivative cell lines of the original frizzled 4 knockout cell lines that are defective in their matrigel behavior. And so here's just a northern blot showing SOX17 expression. Here's the endogenous SOX17 message in a wild-type cell line, and in a frizzled 4 knockout cell line, there's no detectable SOX17 message. And now if we make this stable cell line with lentivirus, we see the lentivirus-derived message at a different size than the endogenous, and at about the same level, making SOX17. So this isn't an overexpression by any stretch. This is basically the normal level of SOX17. But there's a profound effect on the phenotype of these cells. So here's the parental frizzled 4 knockout cell line, and now if you just put SOX17 back into it, it gets up and dances and wiggles around on matrigel and forms these capillary-like networks. And this is true of multiple independent cell lines. In every case, when we put SOX17 back into a frizzled 4 knockout, we restore the phenotype. So we're thinking that this is a signaling system that has evolved a very specialized ligand for essentially a private ligand for this frizzled 4 receptor, and it's funneling the signal through this one particular transcription factor, or at least much of the signal, to activate a genetic program to form capillaries. And we hope that not only will this be relevant to normal development, but it may tell us something about retinal vascular disease as well. Thank you.
It is now my privilege to conclude this wonderful event with a few words in the name of the Champalimaud Foundation. I do it in representation of my president, Leonor Beleza, who had to stay in Lisbon for a meeting of our board of trustees. This unfortunate coincidence of dates did not allow her to be here with us today, but her absence in no way reduces our interest in sharing with all of you our feelings of gratitude towards those who contributed in a decisive manner to make the Champalimaud Vision Award such a successful initiative. The Champalimaud Foundation is a very recent institution. It has a simple and well-defined mission to promote medical research. And although our mission, as defined by our founder, does not restrict us to vision, the Champalimaud Vision Award was our very first initiative, and one we are extremely proud of. From day one, we obtained the irreplaceable support of ARVO. Todd Margolis and Martin Jaeger, current and past presidents, and Joanne Engel, executive director, have been unwavering in their enthusiasm and generosity to help turn a simple project from a little-known new foundation into a highly visible initiative. Without that visibility, our award would not have the notoriety required to bring many high-quality nominations. And it is the quality of the candidates that determines the reputation of the award. This year, we're proud to collaborate with ARVO on a new front. The ARVO Foundation for Eye Research has launched the Host a Researcher Award program to bring investigators from developing countries to the ARVO meeting and to pair them with host researchers in their fields. We were very happy to collaborate in this venture. In a meeting whose theme this year is to reduce disparities in eye disease and treatment, it is fitting to remember that in the current environment of economic crisis, supporting those who can make a difference where care is most needed is fundamental. Working together with ARVO and the ARVO Foundation to enhance capacity building in the areas of the world where it is most needed is a great privilege for the Champalimaud Foundation. I must also add that it is inspiring to collaborate with the ARVO Foundation alongside people like John Dowling, a tireless enthusiast of high research and a great example for all of us. The Champalimaud Vision Award relies on a world-class jury. The jury, chaired by Alfred Sommer, and here most appropriately represented by Paul Sieving, includes some of the very best vision scientists in the world, as well as several personalities from government and culture, all with a long-standing commitment to international work to meet the needs of the developing world. As you may recall from our presentations here in preceding years, and as Paul just recalled, the award alternates between recognizing outstanding contributions to the alleviation of vision impairment and blindness in developing countries, which is done in odd years, and major scientific breakthroughs in the understanding of or preservation of vision, which is done in even years. Today, we are recognizing our first research laureates. This is therefore a very special event for us, and I hope for all of you as well. I cannot add anything to what was already said on the outstanding value of the research contributions of Jeremy Nathans and King-Wai Yao. Paul's observations and everything that was said here today will, I'm sure, stay in our memories for a very long time. In any case, both Jeremy and King-Wai are already so well respected that they would never need any further compliments from us. On the contrary, it is the Champalimaud Foundation that is extremely proud that our award went to such outstanding researchers after a competition that was intense and required hard and serious work on the part of the jury. The jury worked through a long list of well-prepared nominations. Their final decision was reached in a meeting last year in Paris. We follow their labors with immense interest, and we are extremely grateful for the input and enthusiasm they put in the process. The process is very symbolic of how science is developed and research progresses. Our ultimate goal is achieved through the contribution of very serious and respected individuals and institutions who work together for a collective aim, which is of limited direct interest to them. They generate support and visibility for those that are on the leading edge of progress and advancement of science. It is our view that a prestigious award is the least that our societies can do to recognize and thank these great scientists. Without science, we would all be living in a primitive world of ignorance, suffering, and poverty. We will never thank and recognize scientists enough. It is, of course, for this reason that the Champalimaud Award was created. Our founder, Antonio Champalimaud, instructed us to support research. And we concluded that one of the most effective ways of doing so is to honor research achievement, to celebrate great researchers, to present the laureates as symbols of what is best in our societies and what deserves maximum encouragement and recognition. We sincerely hope that by honoring King-Wai and Jeremy, we will perhaps trigger the decision by other young people to go into science, stimulate other young researchers to continue their efforts with renewed energy, and convince every current and future researcher that scientific achievement does have visible compensation. Let me also add a word of respect for our founder, whose decision to create the foundation is also highly symbolic. He was not a man of science. He was a businessman who, through extraordinary entrepreneurial achievement, created enormous value, provided jobs and prosperity for many people in several countries of the world, and built up a large fortune. Yet, at the end of his life, he chose to recognize science as the most deserving goal for the use of his fortune, and generously contributed all he could towards the simple mission of supporting medical research. In our materialistic world, philanthropy represents one of the most heartwarming and at the same time powerfully effective endeavors. Without philanthropy, scientific progress would be so much harder, and we would all be so much poorer human beings. This year, the award will go again to an institution that fights blindness in the developing world. The recipient will be announced in the fall, and we already have a large number of candidates. But I'd like to conclude by reminding all of you that as of October, we will be expecting applications and nominations for next year's award, which will go to scientific achievement again. All currently active and productive groups in vision research whose work deserves world recognition can be nominated. The award is designed to further the resources of their laboratories and can be used in any way that helps their research goals. And all of you in this room surely know those who deserve to be nominated. We're very hopeful that yet again the jury will have a hard time choosing from a vast number of outstanding candidates, and that another King Y or another Jeremy will be here in 2011 to be recognized as a great scientist. Thank you.